All right, so today we're going to be talking about cell structure and function. Here's some of the SLOs we're covering. The two blue ones are review, so cell theory, microscopy, we've talked a little bit about before. Do you want to give a quick breakdown of some of the other important parts that we're going to be
Right,
looking
so
at?
we're going to now cover membranes, a little bit of molecular transport, and then dive deeper inside the cell to look at all the organelles, at least the key ones we're interested in. And then at the end, a little bit of an introduction to classification.
All right.
So where we started is from the last couple of topics, is that we introduced chemistry and we started introducing the use of atoms, and in particular, the one we were interested in was carbon, especially in the organic chemistry side. And we started building those macromolecules, and ultimately, what we want to build is a full-blown cell. But to remind us from last time, too, even from topic A, is that lots of people came before us for the last 400 years or so, and this idea of cell theory dictating that all cells come from originally pre-existing cells, and cells being the basic unit of life. Just a quick review on this is where we're starting, right?
All right, so cell structure. Here we're going to get into some of the stuff that students might remember. The organelles, there's always a few that they remember. Like they always talk about the mitochondria and whatnot. That's like the main one people remember, but we're going
I
to get
have
into.
no idea why they do that, honestly. So let's go first by definitions. First one is two key features of a cell, which are going to be, since they are the basic unit of life, there's two key pieces I want everybody to know. One, that they're self-contained, meaning they have some sort of barrier that contains them or holds them together. This is called the plasma membrane. We'll abbreviate it PM all the time. And then that they're self-sustaining. In other words, they can live on their own. And they do that by actually having those so-called organelles that we talked about before, keeping those alive. So what we're going to do is kind of cover first the outside of the cell, so the plasma membrane, that barrier, that container that we're talking about before. And then eventually we'll highlight the inside, so-called the cytoplasm, which gets divided into the liquid, the cytosol, and then all the solids in there, which are going to be the organelles themselves. So that will come at the latter half of this lecture.
All right.
So,
So talking about the plasma membrane. Um, this is kind of just the skin of the cell, basically. It's made up of these phospholipids. They're bipolar, hydrophilic. They make this like bilayer, right? There's two. They're kind of stacked on each other,
that's right.
opposing.
So those phospholipids that we're interested in, if we learned those in topic B, come with this unique shape in which two of the little fatty acid chains happen to make it very, very hydrophobic. They don't like water like most oils. But then that little phosphate attached to the left-hand side you're seeing in the slide usually makes it hydrophilic. So we end up calling the cell bipolar, not the cell, the membrane. And so because it has these two unique properties in the same exact piece of chemistry, we have hydrophilic and hydrophobic combined, we call it a bipolar phospholipid. However, it's too difficult to know it by that structure, so we're going to simplify it for you guys. And so we end up drawing it like a giant little circle, like a little head, and two tails kind of sticking out from it. And so I've designed already a version of this in which I've colored it in, what is it, red and purple, if you can see in there. And like you were saying earlier, when placed in water, it starts assembling. Now, the unique feature behind this is that, yes, it forms this bilayer, this sandwich, more or less, as a way to hide from water, but it still ends up exposing most of that purple area that you're seeing there from the front, the back, the sides, and so on and so forth. So what do we do is when we see this little bilayer form, it starts extending, it starts getting longer, per se. And so that extension prevents it from getting covered in water from both sides, from the top or the bottom, if you will. But to prevent it from any angle, it circularizes, and then it becomes more like a sphere. So pretty much in all three axes and all 360 degrees, you end up having this bilayer around, which ultimately looks like a little balloon, like a little bubble. And that is more or less the phospholipid bilayer in and of itself. It's a balloon, it's a container, which is one of the properties of the cell. It's a self-contained system.
So that makes up most of the outside of the cell. There's also these little proteins and whatnot that allow nutrients to go in and out and things like that, right?
Right, and so that's where the properties of the cell membrane end up being called a fluid mosaic model, meaning that it's made contains this ability to move, fluidity, and the other part is other pieces that are part of it, hence the mosaic side. Phospholipids end up being the main portion of it. We already discussed that little piece. But then things like cholesterol that we had covered also in topic B, talking about increasing this ability for the membrane to flow or move a little bit more. And then, like you said, proteins, which sometimes can be integrated into the membrane or kind of associated with the membrane on certain angles, inside, outside, that kind of thing. And so those are referred to as integral or peripheral membranes. And not to forget that even carbohydrates are part of those membranes in terms of being at the ends or tips sometimes of those proteins, allowing them to perform certain functions. So overall, it's this mixture of carbs, cholesterol, lipids, proteins, all putting together the so-called plasma membrane itself. Now, depending on where you see this, different textbooks will show you different pictures, different colors, different angles, and so on and so forth. Probably one of the coolest ones that we have over here on this slide kind of indicates how complex it gets with a little bit less color per se. But even the textbook gives you this nice version of it. And as you can see kind of from the right-hand side, our little phospholipid that we created earlier kind of fits in that pattern. Now, what we want to learn about the cell, again, that barrier, is one, it's a barrier, easy, but part two is that it's semi-permeable. The word permeable should come into play over here, meaning that it allows things in and out. And so we kind of have to highlight its purpose. One is separation, meaning it is a container, right? It keeps things on the outside, and things on the inside, very simplistic. But part two of that is that it can control it. The word is regulate in this case, what can go in or out. And that's an entire topic that we're going to talk about in this topic called molecular transport. Now, what does it, allow in or out? Pretty basic here. Anything from nutrients to waste products to building materials, hormones, and so on and so forth, all need to go in and out of the cell. The problem with this is that not all of them can cross. Usually the ones that get a free pass are going to be anything that is tiny, so small molecules or small elements. So your oxygen, your carbon dioxide easily can pass through. And anything that is uncharged, so anything that doesn't have a electric charge associated with it, so not positive, not negative, can also pass through. So things like water sometimes can pass through without any issue. But anything larger than that has a pretty hard time passing through.
All right, so breaking that down a little bit more, molecular transport, we're going to talk about how things move across that membrane.
So we're going to cover several different angles on this. And so we divide it into two main categories, what we call passive and active. Under passive, we're going to cover two key ways of moving things across the membrane called diffusion and facilitated diffusion. And then we always end up talking about the little cousin called osmosis that is still diffusion, but it's usually of water. And the other version is active transport, in which in this case, now it involves energy. It involves some sort of, again, activity associated with it to actually get it to happen. And so that gets covered under what we call direct active transport, which is the one we're going to cover, or large-scale transport itself. Now, I do provide a couple of definitions here that are taken strictly from the textbook, so if people want to read those aloud, they can go and get some additional material there. Now, to kind of explain this a little bit better, it's easier to kind of give you an image of it. So what I'm going to try and do is kind of help people go through this. And the idea is if we were to draw just a membrane of the cell in the middle, right? So here we have the bilayer. We can claim that the inside of the cell is on the left. So this is usually called the intracellular fluid, or ICF. It's an easy way to call that. And the outside of the cell is considered to be the extracellular fluid. And the idea behind this is things need to move from one side to the other. Now again, freely, things that are small or uncharged. Everything else is going to have to pay some sort of price or need a little bit of help. Now things are constantly moving back and forth, but the idea is to hopefully try and balance them out. Something that is called dynamic equilibrium. What that means is that stuff still moves from left to right, right to left, whatever you want to call it, but ultimately they want to kind of end in this state called zero. And what it really translates to us in life, it's homeostasis. Things are at balance, they're even though things are constantly moving in and out. Now, if we take a look at it from the cell's perspective, that's the ICF that we were talking about before. And then we have the ECF on the other side. And what we're going to try and do is demonstrate how things can move from the outside to the inside or vice versa by showing different methods on how to do so. First one behind this list is called diffusion. And so this is by definition things moving through that membrane, through the plasma membrane, in a specific direction. And the direction, this is something to always remember, high to low concentration. Okay? Meaning stuff that is really, really highly concentrated, lots of stuff on one side, is trying to kind of expand and move on to a more dilute or less concentrated area. And again, the idea is to establish that dynamic equilibrium. The concept that more or less, even though things are moving from left to right or right to left, outside in, or so on and so forth, eventually you're trying to reach that homeostasis. Now the key feature here is that, again, this happens freely. There is no energy required. And so anything from basics like gases, like we were talking about, or oxygen and CO2, undergo this particular process. However, in real life, we can actually see this from anything that is highly concentrated. So anything that we detect, like in smell, happens to do the same thing. So perfumes, colognes, and certain sprays do the exact same thing. Areas from high concentration immediately kind of spread out from the area, and then eventually they kind of go away. So the image here is kind of showing more or less both directions, from a high concentration to a low concentration, even though you can even see the arrows moving in both directions. Things are always moving on either way, but ultimately the main drive, high to low.
And this one's just kind of like a law of nature. It just really naturally happens.
Correct. This is good old physics.
I mean, anybody who's into chemistry, I'm trying to remember which gas law it was. Or which law it was, Graham's law or Henry's law or one of those two. But yeah, it's like something that we observe all the time, especially with gases, that they'll diffuse naturally, just try to reach this state of equilibrium.
That's right. And one of the cool things about it is you demonstrate by using cool tricks like balloons and demonstrating colored particles. That happens all the time. So good chemistry classes are kind of actually fun to see. Now, part of that diffusion portion is that sometimes, remember, water can also move. And so when water is the one that moves, we call it the diffusion of water, fairly straightforward, but it has a fancy name called osmosis. Now, I don't want people to get confused over here because it's still a high to low concentration when we're talking or referring to the solute, the material that we want to move. But when the water actually moves, it ends up going in the opposite direction. It goes from the lower concentration to the higher concentration of water itself. Okay. Now, again, this is always happening freely. And since water can pass through the actual membrane itself, then it can just go uncontrolled. It can go from either side, inside, outside, so on and so forth, while other things that are much larger or even charged cannot pass through. So if the solutes can't move, if the big stuff can't pass through, then the water will do it. And so that's actually called osmosis. Now, this is a very, very important topic for us in biology and anatomy, physiology, and pretty much all across anything clinical or medical. Why? Because when we start measuring somebody's homeostasis, it's important to know what their conditions are on the outside of their cells. The term is called a cellular environment. And pretty much depending on what chemistry you might have taken, or even some gen bio, you learn about three key environments called isotonic, hypotonic, and hypertonic. And here a good idea is to pay attention to the etymology of these words. Right, the term tone meaning pressure, very simple. There's the term iso meaning equal, hypo, low, and hyper meaning high. Mm-hmm. But the way to actually remember these, to make sure you don't confuse yourself, is remember, we're talking about the environment, meaning we're always talking about the outside of the cell. So if we say isotonic, means the outside of the cell is the same. If we say hypotonic, meaning the outside of the cell is lower. And if we say hypertonic, means that the outside of the cell is higher. So for that, we have drawn a quick little beaker, and we're going to pretend that we have a cell kind of hanging out in there with double membrane in red. And we can envision some material, the environment, outside, and then what's on the inside. And so whenever both of those, the outside environment and the inside environment, are roughly the same, that's an isotonic condition. Whenever the outside is kind of low, and then the inside is higher, that is a hypotonic environment, so the outside is low. Mm-hmm. And then the direct opposite, lots on the outside, little on the inside, this means it's a hypertonic environment, so the outside is higher. Mm-hmm. Now, why do we care? Well, typically, when we actually take a good A&P class, we end up playing with our blood cells. One of the key ways to examine the health levels of our cells is by taking a little drop of blood and then placing it in all those three environments, an isotonic one, a hypertonic one, and a hypotonic one, and you can look at them under the microscope and actually see what they do in those three environments. Now, logic would tell you that all your cells want to be in an isotonic environment. The same on the outside, same on the inside, so they're all happy. But sometimes the body changes. Diseases can do that. Genetics can do that. And so the cool thing about doing this in a lab is by actually seeing that if you place your cell in a hypotonic environment, for example, meaning the outside is low, the water naturally will rush into the cell, and your little flattened red blood cells that everybody's used to seeing kind of swell up into a balloon and blow up. Whereas if you place them in a hypertonic environment, meaning the outside is high, then the water tries and dilute that, rushes out, and the cell more or less shrivels up and dies. So again, we want to aim for that isotonic level. Outside, inside, roughly the same, so the water, when it moves, it doesn't really change the shape of the cell. So we give them fancy names. So as long as it's isotonic, everybody's happy. But in the event of presenting a hypotonic environment, the cell, when it takes on the water and explodes, this is called a cytolysis, cyto for cell, lysis for breaking up, or it's also known as osmotic lysis because the water is rushing in. And in the event of a hypertonic environment, the cell having far more stuff on the outside than the inside, the water will rush out and the cell will shrivel, a process known as crenation, or also can be known as plasmolysis. So it's important to always maintain your cells in this happy little environment called an isotonic environment.
All right, so moving on to facilitated diffusion.
Right, so now we were talking about a little bit earlier, so things can move, but sometimes they can't. So what happens when you have things that are larger or more or less electric, charged, like ions or something that is ionic? Well, they're going to need a little bit of help. Things are still going to move from a high to low, but more or less still kind of free, but they get an extra little bit of help. So what kind of things could do that? Well, probably the main thing behind every cell is their food, nutrients, and so glucose happens to be number one nutrient on our list. And since glucose is a relatively large molecule, it can't pass through the membrane. So we need to get it somehow into the cell. So it's going to get a little help. Same thing applies for ions or electrolytes. So everybody talks about electrolytes, whether it's an A&P or you're taking a neural lecture, muscles, or something like that. We know that we need to regulate that electricity, but they can't cross. Electric things don't cross the membrane. So again, they need a little help. So what does that look like, more or less, is they get a protein helper, called a protein, more or less, called a facilitator, or hence the term facilitated diffusion. But believe it or not, we get lots of fancy names behind them. You'll hear them called transporters, co-transporters, channels, antiporters, lots of different names, more or less meaning the same thing. It's a protein, it's inserted into the membrane, and it acts like a little tunnel. It lets things in or out that normally couldn't cross through that membrane.
All right, so active transport. And talking about that, does that take ATP? Is that how those pumps work for the most part?
Right, so you hit the key point over there, the term active, meaning it requires energy. But the reason why it's going to require energy is because it's usually going in the opposite direction, something we can call against the concentration gradient. And so that means this motion is low to high concentration, right? And so again, anytime you go against something, you usually have to invest a little bit more energy. So the key feature that you just said there is ATP. In other words, you have to pay for this to actually happen. Now it does come in a couple of flavors, and so there's direct and indirect, more or less. We pay attention to the direct version because that's the easiest one to kind of measure, but there are other ways to do so based on concentrations just without ATP itself. So we'll look at the ATP one first. And so same idea, you'll notice that the diagram shows the same thing, protein in there, but you can see that the direction is now going from a low to high, and now by the green arrow that you're seeing in there is that's the ATP. Because you're going against the natural way of things per se, the way that physics wants things to work, now you have to invest some energy into it. So you have to pay for this to actually happen. Now we're going to take this on a whole different scale now. So far, we're talking about just little pieces of the membrane doing this, but in reality, the entire cell ends up performing these tasks. And so the key feature here is to understand that the whole cell can do this, the entire body. So this is referred to as a large-scale transport. And so that will come into play in cool new names now called endo and exocytosis. To get things in, the word endo here, or the root endo here, the prefix, and the prefix exo, meaning going out. And so the endocytotic events come in two big flavors that we want to cover, phagocytosis and pinocytosis, which stand for cell eating and cell drinking. Now there is a third one that exists that is far more complex for us to cover, so we kind of usually leave it out. So let's start kind of discussing these briefly. Under endocytotic events, the first one that everybody hears about is always cell eating or phagocytosis. And this one's really kind of cool simply because you get to envision the cell itself moving and using that actual membrane as a tool, as an instrument. And so it uses it as if it was creating its own tentacles or tendrils and reaching out to things, grabbing them, holding them, and then bringing them in to eat. Probably the number one example that we always have are your white blood cells, your macrophages specifically. They even show that term phage in there, that means cell eating. And the idea is when they're going around cleaning your blood, cleaning your system, they find bad guys, they wrap around them, and they pretty much chew them all up. Now the other version behind that is more or less called cell drinking. This is called pinocytosis. Sometimes it could be called macropinocytosis. And here, if one of these is cell eating, the other one is cell drinking. And what that means is the actual body of the cell kind of turns and kind of inverts itself or tries to turn itself inward and forms this little bubble, this reverse bubble, and it takes up all the water towards the inside, and whatever's in that water goes in. Now, like I said before, there is a third version, but that is not the subject for us, so we'll leave it out. So here in the image, we kind of display all three. I'm going to erase the one we're not interested in, and you have two versions. One in which you can see parts of the membrane extending, reaching out to grab things and bring them in. And then the other one, you can see kind of the cell diving inward, a process known as invagination, and takes this liquid, and whatever's in that liquid goes into the cell itself.
So a piece of the cell membrane ends up breaking off, huh?
Correct, and those end up forming these organelles or mini organelles called vesicles, the term that probably a lot of people have heard before too.
Okay.
So some pretty little examples of what I meant by those extensions. So you can see the actual plasma membrane in a series of photographs here kind of extending out and reaching out and grabbing bacteria. That's a usual easy way to kind of show that. Here's another better image of showing one entire white blood cell, one entire macrophage, and you can see those tendrils kind of sticking out and grabbing onto things. And then, just for the sake of a good imagery, I wanted to show you a little bit of a video doing this. And so we have the little macrophage kind of chasing around a little diplococcus over there. And on the top image, you can actually see it when it catches it, and it kind of takes it in and swallows it in. But to better understand this, if you can see the other image that we have here, the lower right-hand side, you can actually see the cell making those extensions, grabbing onto things, and then bringing them into the actual cell. And ultimately, you can also see the process of the entire cell grabbing much larger features and just pretty much swallowing them up completely. So these are active events occurring inside every single human at the moment. All right, so the cell drinking version, like I said, it's kind of a divot inward. And so that kind of shows us that the cell kind of turns its membrane towards the inside. And when it wants to bring things in, it forms these cool little vesicles and closes them off, or pinches them off, if you will. And then whatever's in that liquid, it more or less drinks it in. Now, if we want to kind of conclude this, then the reverse of cell drinking is cell, in this case, exocytosis, which is the cell expunging things out. And it's pretty much the exact reverse of pinocytosis. So now those vesicles fuse with the membrane, and whatever makes it to the membrane gets pretty much spat out. So the contents get just thrown out as if the cell was more or less vomiting it out to the containers. All right, so this is a slide just pretty much designed to kind of summarize everything. So we put together all 5 of the main ones we covered that we were interested in. And so we talked about passive versions, active versions. Under the passive, we covered diffusion, facilitated diffusion, and the side version called osmosis. And then on the active versions, we talked about active transport using proteins going in the opposite direction, and the whole cell transport, which were our endo and exocytosis. And so we're going to kind of move on from this, from the movement across the membrane to control what goes in and out.
All right, so we've covered a lot of things on the outside of the cells, the cell membrane, how it moves things into the cell. Now we're going to start talking about what's inside the cell. We're going to break down some of the organelles. And we're going to show some, I know we have a few like generic, very generic images or diagrams of what a cell is, or some of the basic organelles that we know that exist inside cells, but not all cells have all of these organelles, right? I know there's differences between each cell and whatnot, but this is kind of like a generic idea of all the basic organelles that we're going to get into.
Right, so since we're going to cover organelles, right, these work in terms of definition. The term elle meaning small, so little organ or organelle means it is behaving as the organs we have inside humans. Their idea is to keep you alive or performing certain functions. So our organelles do exactly that. Tiny little things that perform certain jobs to keep the cell alive. That's the idea. That's what we want to look at. Problem with it is that anytime we see a cell, it looks like what you're seeing on the lower left-hand side, that it's this giant thing full of stuff, kind of hard to see, very mixed up, and it doesn't really give a good perspective. So instead, we're going to create our own model cell. We're going to pretend that the whole cell is more or less hollow. So we're going to treat this as if it was like a little bowl. And then little by little, we'll cover all the organelles that we're interested in, the ones that you're seeing on the list on the right-hand side, and add them one by one. So one of the easiest ways to kind of learn this and to study is by kind of drawing them separately. In other words, take a little circle, draw it around, and one by one, write a function and write more or less their name, and try and remember them that way as opposed to having this giant image full of stuff. So the organelles we're going to cover obviously are within the cytoplasm, meaning it's the liquid plus the organelles, the cytosol and the organelles. And a good chunk of these are, believe it or not, made up of that same plasma membrane we talked about earlier, the same thing that is on the outside. And so a collection of these is referred to as the endomembrane system simply because they're using those phospholipids as well. So not only do phospholipids play a role on the outside of the cell, but also partially on the inside of the cell. So what we're going to do is look at each one of these one by one by kind of demonstrating their function, obviously their name, give you some images, but also the way I try and usually refer to these is by kind of giving a little metaphor on how this relates to if we were in a business. Like somebody was having a little store, and who runs the show, what goes on the inside, on the outside, you know, how do things ship out, that kind of thing. We're going to do the same idea here. So, like in any business, for example, one of the key ones we always talk about is somewhere where the main office is, where all the big instructions happen, where all the main orders come into play. And so that is our nucleus, right? So that can be considered the head office. Now, our nucleus is also composed from those phospholipids that we mentioned before. And so this is a membrane-bound organelle. And the reason why we consider it to be the main place is because typically this is where we find DNA. In other words, the instructions of all life typically are found inside this container, or something like a container, and they condense up into things called chromosomes. Now, that's not normally the only thing that is inside the nucleus. Sometimes there's this little region inside of it called the nucleolus. The term olus also meaning little, so like a little nucleus. And that plays a role in also building RNA. We'll talk about that much later. And also the production of the second organelle that we're interested in, called the ribosomes. So this is the main place where the instructions begin, per se. Now, if we wanted to look at them under the microscope, typically speaking, it takes a large amount of volume. It's typically very central, or in the middle of the cell. And most scientists, when we actually play with these, we like to stain them bluish. Not always, but a good chunk of the time, as a way to identify a nucleus, under pictures, under imaging, we stain these blue. Now, I did say that the second organelle we're going to look at are the ribosomes. This is present in every cell that we know of. And if we have a boss, like the nucleus, barking the orders, we also have the workers, somebody that actually does the job, that performs all the tasks that are needed for the actual cell. So our ribosomes are our workers. And so these guys are tiny little organelles, kind of difficult to see under the microscope most of the time. And they're made up of two things, protein and RNA, so a little bit of a combo. And so they come in two pieces, two subunits, one large one, one small one. And their sole purpose, like most workers, is to build product, to make something. And so that is called translation of proteins inside our cells. They build proteins by literally following the directions of the boss, meaning the DNA, and taking amino acids one by one and assembling them through dehydration synthesis to build polypeptides or proteins. Now, what's interesting about this is that these ribosomes can be found in two specific locations, typically kind of freely floating around inside the cell itself, so in the cytosol, or you can usually find them in a workplace, a location called the endoplasmic reticulum, our third organelle that we're going to see in a moment. Now, the issue with ribosomes is that they're so small that they're really difficult to come up with an image. We have barely enough technology to do so. And so most of the time when people show ribosomes, they show computer images of them, models of what people think, and that's what you're seeing here. But on the lower right-hand side, you're seeing scanning electron micrograph, an SEM, of one little piece of RNA with tons and tons of little ribosomes attached. So as you can see, they're really, really small. Makes it difficult to see. But with our latest technology, we can actually take partial images of these guys, as if we were taking like little pieces of a movie, and then reassembling it to give us a three-dimensional view of them. And so that's how we know more or less the rough shape of a ribosome, and that it has two pieces. So that's kind of how we end up seeing what you're seeing here on the image.
That's pretty interesting.
Right, now, I did say that you can find these guys floating around, or they'd be attached to a third organelle. This is called the ER, most commonly abbreviated that way, or what most people know as in the endoplasmic reticulum. So this is the actual work site, and it is also part of that endomembrane system because it's also made out of phospholipids too. And this is a fairly large organelle, so it's all large membrane-bound little packets, or in this case, layers of plasma membrane inside, and it comes in two shapes also, two flavors, if you will. One of them is very kind of smooth on the outside. You can tell the difference immediately. And this location is solely designed to produce certain hormones, as well as to help the cell break down certain things and clean things up, especially lipids and fats. But there's a second version where those ribosomes that we just finished talking about are attached. And so because they're kind of embedded in the surface of the endoplasmic reticulum, it ends up looking like little bubbles. And so that section of the ER ends up being called the rough endoplasmic reticulum, or the rough ER. And so this is also where work is happening and we're producing protein. So overall, understand that the ER performs two separate, very distinct functions. One, yes, it's the production of protein, just like we mentioned before, but the other portion performs additional functions for the cell, like cleaning it and creating certain signals and hormones. Now, if we were to look at it, again, using the textbook and some image is there, we see, for example, the nucleus kind of in this purple color that you're seeing over there, but then you see this continuous membrane associated with it. And so you can see two versions of it. One that looks like it's covered in little dots, and the other one looks significantly smoother. And so the smooth ER in this case is usually afterward, whereas the rough ER is usually closer to the nucleus, kind of keeping the process of production close to the nucleus itself, reminding you that that's where the instructions are coming from. Now, if we look at this under the actual microscope, so this is using an SEM, so scanning electron microscope, you can see the nucleus at the bottom, and then you can see all these sorts of little membranes kind of spread out everywhere, and you can almost see the graininess of where each one of the individual ribosomes is attached to that membrane. So every single dot in there where it says the rough ER is where proteins are being made. So all those little factory workers or ribosomes are attached in there. But if you look a little bit further outward towards the top of the screen, you'll notice that that membrane gets smoother. And so that means no more ribosomes there. That's our smooth ER. And again, an entire different set of functions here. Here you're talking about detoxification, cleaning, fat degradation, and even the production of certain steroid hormones. Now, that being said, there's more membranes And then the next one in this order is called, under different names, could be called the Golgi apparatus, Golgi body or bodies, or also called the Golgi complex. And also part of that endomembrane system. It's still made up of phospholipids. It's still a relatively large packet of membrane, more or less. But here, if we have a business like we were talking about before, most of the time after you're done creating your product, whatever this may be, you still have to kind of package it, label it, know where you're going to send it, how to distribute it, and so on and so forth. And that's more or less what the Golgi does. So that SNH that we have on there symbolizing shipping and handling, the idea is you are preparing things to be delivered to the right place. So what happens in the Golgi is after the proteins are made by the ribosomes, either freely floating in the cell or at the rough ER, they get activated, they get turned on per se to do their job, and then they get shipped, they get transported to the ultimate destination, towards where they're going to do their final job, more or less. That's what the Golgi apparatus does. Now, if we look at it inside the cell, though, same idea. It's a bunch of membrane, kind of relatively close to the nucleus also. And if we look at it under an SEM, you can also see the nucleus, and you can also see those little membranes kind of relatively close, ready to receive protein, ready to activate them, turn them on, and then ship them off to where they need to go.
All right, so I like this factory analogy because it all actually kind of makes sense. So we talked about the nucleus, that's where all this information is held. We've talked about ribosomes, those are what are building things. And we've talked about the endoplasmic reticulum, also building some things, working with the ribosomes. Golgi apparatus, that even goes back to, we learn about the powerhouse of the cell, but the Golgi apparatus is kind of tied to shipping and handling kind of areas. Now we're moving on to lysosomes. So those are cleaning crew and recycling.
Right. So like any other business, also made up of membranes in this case, we need a place to clean up. We need something to help us kind of remove any wastes, to clean up any damage or anything like that. And so these are smaller versions of the same things we were talking about before. So it's still phospholipids. But in this case, when we talked about earlier about cellular transport, endocytosis, so phagocytosis and pinocytosis, bringing things into the cell, there are these little vesicles called lysosomes, also membrane bound, again. They're designed to more or less clean things. So they break down any trash, they remove anything that is extra. But what's really cool about this set of organelles is that they recycle also. In other words, not only do they clean up stuff, but anything that can be reused, they'll leave it inside the cell. So the idea is they can even consume a bacterium, which is what you're seeing over here, and they'll blow it up, clean it up, and anything that remains in there, certain amino acids, certain nucleotides, whatever it is, they'll actually keep them inside the cell for later reuse. So not only is it a cleaning portion, but also a recycling portion behind it. And so you can see a cool transmission electron micrograph, a TEM, of those little vesicles on the right-hand side too. Now, talking about these inclusions, these things that are inside the cell, like little membranes, this is a really big one. Vacuoles, also part of the endomembrane system, act like a trash bin. There's no better way to say it. This is where all the bad stuff usually goes when we don't know where to throw it away. So it's a sewer. And so these can get fairly big. They can take up to 50-70% of the body of a cell. And usually they take up a lot of volume simply because they're full of water. This occurs through osmosis. And so most of the time they can store up certain nutrients, certain products, but most of the time they end up storing waste. So this is a way to throw trash and keep it from accessing the rest of the cell because they could be toxic. And then eventually, once the cell decides that it wants to get rid of it, it goes through that molecular transport that we talked about before, and more or less exocytoses it out or vomits it out and gets rid of it for the cell, and it's nice and clean again. So now what's interesting about this though is that vacuoles typically are only found in plant cells, couple of fungal cells, and some weird little algae-like organisms. You don't typically find these in humans or in animals most of the time. So this is very common for certain other types of organisms. Now, you were talking about this, so I'll let you go off on this one. I'm sure you can tell us more than that.
All right, so the mitochondria. So this one's kind of interesting. There's a lot of complexities within the mitochondria. The most basic thing is people know it produces power, like it produces ATP. That's the basically the little batteries of the cell that get used throughout to perform functions. How it produces it, we're gonna really break down like way in the future, but and it's really interesting actually. But basically, it gets energy from glucose. We'll talk about that whole process, glycolysis, where this whole process happens that breaks down sugars and produces energy, and that get used around the cell.
So that process that you just mentioned is one little piece of the whole thing called cellular respiration. So glycolysis plays that role. There's more in there. And more or less what it's doing is taking some sort of nutrient and turning it into energy. So this is why everybody, whenever they hear it, they have that classic term of powerhouse of the cell. Nobody can get over it, pretty much. So we'll keep it as that. So think about this as a nice little organelle that has two membranes, though. This is very unique about it. So instead of having one layer like everything else, it's a double membrane bound organelle. And also because it's kind of tiny, usually it's very difficult to measure or observe. But we do have some SEMs and TEMs that kind of show us that double membrane system. There's usually one layer on the outside, one layer on the inside, and it's between those two layers in which that glycolysis you were talking about, that respiration that we're about to talk about later on in class, more or less is taking place. Food is going in there, we're turning it into energy. That's really what this guy does. Now, if you're already familiar with the mitochondrion, which again, everybody usually is, then we're going to kind of one-up it. We're going to, instead of talking about two membranes, we're going to talk about three membranes now, and pretty much doing the exact same thing. It's still going to produce energy, still going to make those batteries that you were talking about a little bit earlier, but there's one key feature that changes. Rather than using food to do this, it uses sunlight or some sort of UV light or some sort of radiation to do so. So the key feature here is that if one of them was a powerhouse, then this one has to be some sort of solar panel to keep also producing energy. And so the key term that we use here, instead of calling it cellular respiration, we now call it photorespiration to mean roughly the same thing, but by using light. Now, one of the other indicators on how to relate this to classification is that not all organisms have chloroplasts. Pretty much everybody learns this, even in usually K through 12. We talk about plants having chloroplasts and them being able to produce their own food and energy and so on and so forth. So definitely humans know, definitely animals know, but certain specific cells out there, certain organisms do carry these guys. What's always unique about them is their color. So that triple membrane system that we're talking about, within it has this very unique compound that looks very much like hemoglobin, believe it or not, that traps also iron, but in this case it reflects, rather than red like it does for us, it reflects green. So now most things that carry chloroplasts end up looking green like our plants normally do. So the key feature to always remember here is that most cells will have mitochondria, but the cells that have chloroplasts present, it's not mutually exclusive. It's not that they only have one or the other, they can actually have both. Okay, so before we kind of finish off, the last couple of set of structures happen to more or less kind of emphasize that when people learn about cells is that they're not really hollow. Cells, just like even humans, have a skeleton. They have an internal structure, something that holds them together. And so here, this is called a cytoskeleton. And the easiest way I can usually relate to this, like a building was, something that holds it together, like bone does, for example, inside a body. There's a certain set of proteins called tubulin, and they assemble themselves into these very long strings called microtubules that are long, that hollow. They're like tubes, hence the term, but they're very, very rigid. They're very, very strong. And so kind of like our bones, they're meant to kind of hold us together, give us shape. But if those were to act like bone, then we have actin, which is also another protein, much smaller, also assembles into little fibers. But in this case, it's very bendy, it's very flexible, much softer for it. It stretches out a little bit too. So if our microtubules were to represent our bone, microfilaments end up being kind of like cartilage to give us some flexibility and mobility. And so again, to re-emphasize the fact that our cells are not just a bunch of liquid floating around, our cells are full of these microtubules and microfilaments. They are everywhere. Every cell that you see has inside of it a combination of actin, tubulin, all spread out like a little mini skeleton holding that cell in that particular shape. Now, if we were to kind of look at them a little bit more up close, you can see that actin is a little bit thinner, smaller, tubulin is a little bit bigger, wider, but more hollow. And if we were to look at it under the microscope, under an SEM level and TEM level, you'll notice that really it's very dense, almost like a spider web, almost like these long strands all over the cell that not only give it shape, but even at times they act like little highways. They can allow certain things to be transported through them and ship them from point A to point B.
All right, so we're going to talk a little bit about motility, kind of like getting into how cells can actually move around. And this is kind of fascinating to me because it's like this complex little motor-like feature that cells have. This there's cilia and flagella. So cilia are multiple little hair-like features coming off of the cell. There's short and there's many of them. And then the flagella, longer, there's fewer of them. And they basically work as like tails that are just constantly moving and propelling the cell around. And it's pretty interesting to see, especially if you have the opportunity to see it under a microscope.
Right, so what's really cool about them is the fact that they're built up of those microtubules that we just finished talking about. So what it is these extensions are part of the plasma membrane, and so part of those tubulin proteins and microtubules extend out of the cell, and then they get coated by the plasma membrane, and they act, like you said, like little motors. And pretty much everybody's kind of familiar with cells moving. You can even see them in classic videos. But if anybody talks about AMP, the longer versions, the flagellum version of them, or plural flagella, is what everybody sees in the sperm kind of swimming around. Or if you were to look at cilia, there's two classic ones that we see in humans. One of them is in your lungs. So anytime you're kind of coughing up certain things, things are accumulating within your lungs and you need to cough them out. Cilia work like a little ladder system and kind of bristle them out and sweep them out. Or even roughly on a kind of monthly basis, the transport of the egg, the ovum during reproduction, kind of does the same thing. That egg is transported by little cilia and move it around within the body too. So some images to kind of highlight the differences between short and many versus long and few. So the last little piece that we're going to talk about, and this one we're going to dive a little bit deeper, simply because it plays a much bigger role in bacteria more than anything else, is the wall. And so since we're still kind of alluding to the business, here, if you were to have a house or a business or whatever you want to talk about it, sometimes it's important to bring an optional fence. In other words, not only the walls that surround the actual building, that would be your plasma membrane, but then you can add another wall, an extra outer layer of protection. And so an optional fence, not all cells have them. And so this is referred to as the wall. And so most of the time, this is composed of a combination of carbs, sugars, and some protein. And virtually every single cell that we know of, except animals, usually have these. So your plants have it, and this is the form of cellulose. Shrooms have it in terms of protein called chitin. Protein and sugar called chitin. And then all bacteria have it. This is your classic known as PG or peptidoglycan. And even things like algae have it. But humans, animals, we do not have that extra wall, which is kind of important to actually know, because in humans, one of the biggest advantages that we have as animals is that our cells don't have that restriction. Cell walls maintain size. Our cells can get bigger, they can move more, so therefore we got to be the bigger, stronger, faster organisms as opposed to our plants, shrooms, and things like that too. Now whenever we look at them, though, you can see it from the images here, is that we have our plasma membrane, and usually outside of it you have this thick layer worth of sugar and protein. That's that peptidoglycan, that carbohydrate and protein combination that we have on the outside. And virtually anything from algae have it, which is you're seeing on the lower left hand side. And you can even see things like yeasts having them on the right hand side. These are cells that have an extra layer of protection outside of them. Now, why do we want to actually learn this is because in the actual lab, we get to actually measure this. And so most people that are taking microbiology end up learning a little bit more details behind this simply because they do the so-called gram stain. So that wall that we end up talking about is, again, an extra layer outside of it. And so it consists both of the plasma membrane and the wall put together, ends up being this entire system called the envelope that protects the entire cell. But more or less, it's a layer of material that is designed to hold the shape, to also control how much water can go in and out, and give it shape altogether. But to remind you, it's made up typically of carbohydrates and protein. And so this term peptidoglycan also can be called murine in case you see certain books. And it's a combination of sugars and protein. And so the lower left-hand side, we have a couple of those examples of the two sugars that it's composed of. You'll hear them called NAG and NAM probably most of the time. And then you see on the right-hand side a set of 2 amino acids and 1 double amino acid on the lower right-hand side. And to actually make this a little bit easier to kind of observe, we can just kind of give them some simple shapes, the hexagons and that little kind of squiggly shape. And so outside of the cell, these sugars and amino acids assemble themselves in this kind of fence-like system. And so that is the peptidoglycan layer. And usually you can even add some extra additional macromolecules to give them extra flavors. To simplify this, we're just going to kind of draw this out like a little plasma membrane that you're seeing there in red and purple. And then the peptidoglycan, that wall that we were just talking about before, is that kind of green and orange version of it. And so in the lab, when we learn this, we end up testing which version of the wall they usually carry. And it more or less comes in 2 big types. You're seeing them right now. I'm going to label them on the bottom so it kind of follow along. And the idea behind them is that one of them usually is very simple. It only has that thick layer of peptidoglycan, which is what you're seeing on the left-hand side. And it actually has a little bit of this lipid called the teichoic acid, which is kind of extending through it. And that's pretty much it. It's very simple. Peptidoglycan, teichoic acids, and that's about it. But in a secondary version behind this, you'll actually see the wall that is much thinner, but then there's a second membrane outside of it, an additional plasma membrane. So this is a double membrane system, okay? And then outside of that, then there are certain types of lipids sticking from it, certain types of sugars sticking from them. So it gets a little bit more complex. Now, the big deal behind this and the reason why this is important to us is that these 2 differences in walls lead to different types of stains. When we perform the gram stain, what ends up happening is that the one that is just the peptidoglycan, the one on the left-hand side, the one that just has the little green and orange that you're seeing there, takes up stain like no other. It goes inside of it easily. And so when we're doing the process, that blue, our primary stain that goes in there, gets absorbed, no problem, no questions asked. But in the second one, the right-hand side membrane, it doesn't take up the stain at all. 2 membranes plus a wall doesn't really let much ink in it. So what happens? The ones on the left get automatically stained blue. The ones on the right don't really get stained at all. So during the process of a gram stain, then you fix that color by using iodine or something called a mordant, and it kind of fixes it inside, so the blue stays trapped. But then you add some alcohol, and the alcohol has a tendency of removing membranes. And so what will happen is the alcohol will wash off part of that layer of the second membrane on the right-hand side and leaves it vulnerable to a second stain, a counter stain or secondary stain. And so in the gram stain, that's usually that red-pink color. And so now, the first set of cells got stained blue and are locked in by iodine, but the second set of cells, after you wash them, will turn, in this case, reddish, pinkish color. So what the gram stain more or less does is differentiate between the 2 types of walls. The thick one, usually easily stained, gets hit by blue, and then the double system ends up getting broken down by some alcohol, and then ends up taking later on the color of reddish, pinkish color. So what the gram stain is designed to do is to test which type of wall you have. And that becomes a serious important concept to learn in medicine, because depending on which wall you have, you know how to treat your cells. In other words, you know how to diagnose and treat. Cells that take up the stain will usually take up medicine very quickly. Cells that don't take up the stain, like our gram-negatives, which are on the one right-hand side, usually involve stronger drugs. So this is part of the clinical application that we have over here. So this is kind of important to know. So leaving that last organelle, the wall at the end, the last thing that we need to cover in this topic is more or less just based on these organelles, how do we classify some of the cells? And believe it or not, it's classified very quickly by using the nucleus. Cells that have it are called eukaryotic. Cells that don't have it are prokaryotic. Your prokaryotes versus your eukaryotes. The problem with this is that that's pretty much it. We end up classifying things based on what they contain, so their structures, and there's two basic ways to do so. One is by looking what type of DNA they carry, what type of nucleic acid is, and what's outside of them, the membrane. And remember that wall gets to play part of that role. So what we're going to learn here is by looking at the nucleus inside, where that nucleic acid is, and then looking on the outside too. So whenever we learn about things, we always pay attention to DNA. That's going to be one of our main features. And then the outside layer that protects it, more or less the plasma membrane. And depending on which level of courses you're taking, you can start assembling those larger and larger, more complex systems. But ultimately, no matter how large the actual organism is, they always get brought down to that last or more simplistic system of cells and nucleic acids. That's the way we classify everything. Now, everybody learns about this by learning the old classic 5 kingdoms of life, which we now know to be very inaccurate. And so that's been updated to the 3 domains of life. So in topics F later on, we'll actually spend more time talking about these in detail, just right now to kind of highlight some of these. The old 5 systems come into 1 prokaryotic kingdom, 4 eukaryotic kingdoms, all based on what they look like, what is similar about them, and a little bit of their differences. Now, we've corrected that to include the 3 domains of life, and those are all just classified based on RNA, believe it or not. So we look at them really quickly. The first domain, which is bacteria, the second domain, which is archaea, all fall under the old system of prokaryotes, the no nucleus cells. And that used to be called Kingdom Monera, which are all single-celled organisms, pretty much no organelles inside of them with the exception of ribosomes, and a couple of other weird things here and there. And those used to be called archaic bacteria and eubacteria. Now we've renamed them to domains archaea and bacteria. Now, as far as these guys go, the differences between those 2 domains is that archaea still exist, but usually exist in places we normally don't find very friendly per se. They're extreme environments that they've adapted to live in, and bacteria usually live in ours. One of the key features here is that because they're with us, they usually have a tendency of living on us also and could possibly cause disease. However, under the microscope, not much difference that can be observed. Even when we look at them under colorized and even closer conditions, unless you're an expert, these are hard to kind of tell apart. On the other hand, the last 4 kingdoms, which are the one single domain now, are the ones that contain the nucleus and most of those organelles. And those used to be the old 4 kingdoms: Protista, Fungi, Plantae, and Animalia. And just to kind of highlight some of those features, Animalia, which are the animals, these are all multicellular, no walls like we mentioned before. That's one of the unique features behind them. And they have virtually all of the organelles, and they survive by eating other things, a term called heterotrophs. And so this includes all your animals, including us. But fungi, on the other hand, also happen to be multicellular. Some versions, like yeast, can be unicellular. They have a wall made out of chitin. They have all the organelles too. They're also heterotrophs, but they thrive on surviving on things that are either dead or dying, which is usually where you find these organisms. And most of these include things like yeasts, which anything from producing beer to also causing certain diseases, and your classic mushrooms that you see all in the wild. Those are all under that category. The last 2 include the plants, Kingdom Plantae, also multicellular, also have a cell wall, but that's called cellulose. Also lots of organelles, including chloroplasts. That's where that comes into play. And they're usually autotrophs simply because rather than surviving by eating other things, they can actually produce their own energy using sunlight. Those chloroplasts come into play. And this includes anything from your classic grass and trees and everything else that most of you are familiar with. The last one is the weird one, and this is kind of really where we kind of close shop. It's because protists, the reason why this has been reclassified is because it's everything else that we didn't know how to classify. We kind of found this final group and said we're going to throw them all in there because we don't know what to do with them. And we more or less classify them based on how similar they were to the other three. So if they behave like animals, we call them protozoans. If they look like plants or behave like plants, we call those algae. And if they had properties similar to mushrooms or fungi, they were called moles. And this is a very, very large group that includes millions and millions of species that are very difficult to kind of classify. So we'll take at those one at a time. So that's pretty much our story. So we've kind of gone through several different layers of the cell, right? We went on the outside, we went on the inside, and now we looked at a little bit of classification.
All right, so that pretty much covers everything for this topic. And yeah, it's a lot, quite a bit to take in, but it's exciting. Like, everybody likes the organelles, I think. It's fun to learn about all their different functions. And that is pretty much everything for this one.